Okay, we back to the talk again. So I mentioned one other thing which is important are the previous exposure. Previous exposure, it means either it is the first time or the second time. If it's the second time, it's previous exposure. So as you can see, if the antigen activates the B cell response is different when they exposed for the first time or for the second time. It's completely different. You can see it, the shape is different. So in the first time, when the B cells activated, they produce a lot of IgM and just a small amount of the IgG, but mainly is IgM. So in the first response, mainly we have just IgM, but a small amount of IgG. And it takes about 10 days to immune system to become fully functional. That's why when people get sick and if it's a viral infection, when they go to the doctor usually tells them, go to rest for a week. Why? Because after a week, the level of the antigen will increase and these antibody will eliminate the pathogen. So after a week, the patient will be okay. So don't need any drug for virus infection. It's the reason, because after 7 to 10 days, the peak of the immunoglobulin production happened, and then the immunoglobulin produced, they eliminate them. And little by little, the patient become well. The only thing remain after that is some memory cell. So all the plasma cell will be die, and only memory cell stay in function. But if the same patient infected with the same antigen, it is the second time which they activate the memory cells. And the memory cells quickly produce a lot of antibody. You can see that it is just in 3 days. They produce a lot of antibody. And this antibody not only is huge, but also have a high affinity, is IgG. They can bind and kill much faster than IgM. So that's why the patient even didn't realize they become infected. Because before the symptoms happen to the person, the antibody will eliminate the pathogen, and that's why the patient never feel it. So that's why the secondary response is much higher in the term of the production of the antibody and also the effective the antibody. Because the IgG they produce is more effective than IgM. So that's why they kill and eliminate all the pathogen very quickly. And finally, in the end, you still have the memory cell. So these are the difference between the primary response and secondary response. You can read it. I'm not going to spend here. So through the course, we talk about the interaction between the two receptors. Instead, CD40 ligand, they bind with each other, or B cell receptor bind to the antigen. All these connection give some signal. So it is the surface of the cells which they bind with the antigen. This is what happened inside the cells. It is what happened inside. So any signal goes to this receptor, any receptor, either by antigen or by ligand. They give signal, and the signals are completely different. I'm not going to go through all these signals because it is huge part of the immunology. But what you should know that this signal activates a lot of protein here. They phosphorylate them. There is more than 100, 200 protein here that can be activated. It's based what kind of signal and which receptor is activated. So each receptor actually phosphorylates different protein. So that's why the signals are different from one to another. And then these protein activate the adaptive protein. What these section do, they join the protein to the enzyme here. And when enzyme activated, they activate transcription factor. And the transcription factor, what they do, they give a message to mRNA to produce the protein is required. So if the T cell gives the signal to the B cell, they produce interleukin-2, which is very important for the survival. If, for example, if the fast or fast ligand interaction happen, they give a signal, they kill the cell. So it depends what kind of signal they receive on the surface. The product in the end can be completely different. Why? Because they involve different protein in this interaction. Each signal has a different outcome. Yeah, each signal have a different outcome. The reason are different because the protein are involved is different. I'm not going to involve all. If you just imagine, if I involve to this protein, there is a lot of names, there is a lot of interaction, which is not suitable for you. But if you want, you can go to read it in, some of them actually are in the Molecular Immunology Abu Abbas, the big book. If you read the big book, sometimes they explain some of these signals. So the interaction of you have to know that the interaction of the two cells together, they produce different product. Why? Because they involve interaction of the different protein inside the cell, and this interaction of the protein make the cells produce different product, which is required for their function. That's why when these signal happen, they produce something different, and when two other signal happen, they produce different. Why? Because they involve with different protein inside the cell. For protein antigen, they receive one signal from the antigen. The B cell receive one signal from the antigen, and that signal is not enough. If just the B cell receive one signal, they become energy. Energy means they don't respond to anything, become quiet. So to begin function, they need more than one signal. That's why protein antigen need T cells to receive the second signal or the third signal. So one signal from antigen in protein case is not enough for activate the B cell. If the B cell wants to activate it, at least they need 2-3 signals. Where this signal is coming from? One is coming from so these are the these are the receptor on the B cells, and these are complement. CD40 ligand on the T cell, CD28 is on the T cell. Yeah, all these is B cells here, B7 and 1 and 2. And also we have toll-like receptors which bind with different part of the microbes. So the toll-like receptor are the receptor on the B cell which are able to bind with the microbe. The number 2 is bind with the BLP, which is lipoprotein, bacterial lipoprotein, and toll-like receptor 4, for example, they bind with the LPS. What is the LPS? The LPS is the cell wall of the gram-negative bacteria. Yeah, because the cell wall is have lipopolysaccharide, which is LPS, and or toll-like receptor 5 bind to the flagellin, which make the bacteria to move, and toll-like receptor 7 to RNA, and toll-like receptor 9 to DNA. So for the activation of the B cells in the term of the protein antigen, they need more than one signal to be activated, and the signal is coming from here. It is on the B cell side, and this one is in complement and T cell and from the microbes. CD3. Complement is the protein inside the body. Is Dr. Imad mentioned to you? Yeah. At least 2 signals, at least. It depends on the antigen, and yeah, sometimes they need more than 2 antigen to be functional. Okay, function of, so what happened when the B cell activated? When the B cell activated, they increase the number, and the same time they express the receptor. So, at the same time, when the B cell become activated with antigen, they increase the number and also increase the receptor to be helped by T cell. When they express the T cell receptor, the receptor, the T cell will help you. For example, this receptor mainly is IL-2. So when the B cells become activated, they increase the number and increase IL-2 receptor, which produced by T cell. That's why the T cell can help the B cell to increase the number. And also they produce IgM. It is the interaction between B cell and T cell. So in the beginning, when the B cells present the antigen to the T cells, the T cell doesn't have a ligand here. But when they present the antigen, the CD40 ligand appear on the cell. So if the T cell is not activated by antigen, they don't have a ligand. When they express it, when the B cells give the antigen to the T cell, when the B cell give antigen to the T cell, they
have a ligand.
they express ligand, and then this ligand give another signal, the second signal. And the third signal coming from the cytokine. And finally the B cell become activated and differentiate. So in the beginning, the T cell,
only express and
no, they don't do anything. It's still there. So when the B cells become activated with antigen, they process the antigen and give it to the T cells. When they activate the T cell, the T cells express CD40 ligand, and then another signal come with, and at the same time, they produce cytokine, give the third signal, and finally the B cells become differentiated to plasma cell and membrane.
if there is no ligand, it will be not activated.
If it's not activated, no. The T cell doesn't have that ligand. They have to be activated. Because all this receptor is not expressing on the cells all the time. They come and go. They come on the surface when it's required. When the job is finished, they internalize inside. Some of them, not all of them, but some of the receptor is there all the time.
If there is a defect on the ligand,
No, it depends the activation. In what kind of signal they express the relevant ligand. Okay, the importance of the CD40 ligand is the immune system is very smart and very well organized. If something goes wrong, the person should be in a big trouble. But as you know, most of the people don't have any problem because they do their job very well organized. So what is the important function of the CD40 ligand? So they give the second signal, they cause proliferation. They also make the T cell to produce the cytokine. They make the germinal center and also exocytosis. So if this interaction doesn't happen, none of these will happen. So as we mentioned before, if the mutation here in CD40 ligand, you have X-linked hyper IgM syndrome. It is exactly what happened in the lymph node for production of the immunoglobulin. Question? Yeah, that's why I'm going to that now, little by little. It is exactly what's happening in the lymph node. So in the lymph node, the antigen will be present to the T cells. Can you see it on the top? Because the pointer is not working. So the antigen will be presented to T cell. T cell increase their number, and then they attract the B cell to come to see it. So when the B cell come, they present the same antigen to the T cell again, and the T cell activate the B cell, and the B cell number will increase. Yeah, coronal expansion. The B cell increase. Some part of these B cells. Some part of the B cells produce short-lived plasma cells. They just live 3-4 days and then they die. They call it short-lived plasma cell. Mainly they produce IgM. And a small amount of the IgG. So the first signal they receive on T cell, some of them become short-lived plasma cell, which mainly produce IgM and a little bit IgG. And they call it short-lived because after 2-3 days they will die. They provide the first help to the host to confront it. Because for production of the IgG, you will see it takes time. So to fill the gap, these short-term plasma cells produce a little bit IgM to help the host, and in the meantime, some of these B cells go to the follicle, the follicle around the lymph node, they make a germinal center here. It is in the germinal center which they produce long-lived plasma cells, produce a lot of IgG, and also they make memory cells. So some of the B cells produce short-lived plasma cell to produce IgM to fill the gap and give the time for germinal center to produce the IgG later. So it is in the germinal center that the IgG will be produced. So it is a difference between the plasma cell produced in the short-lived and long-lived. So the difference is here. You can go through it. It is the germinal center. So when the B cell go to the lymph node, here in the bottom, they call it dark zone. They increase the number. From one single cell, they produce more than 5,000 cells very quickly. So all these cells, when they produce, they have to produce receptor as well, because the receptor have to be released to be antibody. So they produce a lot of receptors here, and then when they go to the another side here. They call it light zone. Light zone, the dendritic cell and T cell is available there to help them. And when they get help, they become plasma cell and memory cell. So what kind of help they're going to give them, I'll tell you later in the next slide. So they go to the germinal center. In the dark zone, they increase the number, and then they go to the light zone to see the T cell and the dendritic cell. If they don't see them, they will die. If they see them, they will survive. And then they go to produce plasma cell, which produce the IgG or memory cells. No, short plasma cell is in the extrafollicular. It's not in the, short is here, it's in extracellular branch, a T cell area. This is the T cell area, this is the follicle. T cell area is here. Follicle is here. So it is the germinal center. So the germinal center produce memory cells, and memory cells go to blood, and the blood go to the bone marrow. Yeah, it is how the B cells present the antigen to the T cells. Like antigen presenting cell, macrophage, and dendritic cell. They take the antigen, they take it inside, they break the antigen, and present it to the T cell in the form of the peptide. So that's why they activate the T cells. So B cell and T cell recognize the same antigen in the end. All right. Okay, I explain this one first because I don't know why it jumped to here. I explain this one first and then back to the other. So it is what's happening in the germinal center. So, as I said, in the dark zone, the number of the cells will increase, and some of these cells, they express the receptor, and some of them doesn't. And those who doesn't express the receptor, they will die because they cannot get any help from the T cells. As you can see, the shape of the B cell receptor is different. You can see it. If you compare these three, the shape is different. To understand it better, I'll give you an example. So just imagine the talk ended to 3 o'clock. Everybody will be hungry. So when you go out, you smell food. You go toward the food, isn't it? But some of you might be, your nose will be blocked. So you cannot smell it. So you cannot go toward the food. You don't know where to go. But those people who can smell it, some of them are stronger and faster. Just imagine if there is a small limited of the food there, who get the food? Those who are faster and stronger. Exactly is happening here. If they don't express the receptor, it is those like doesn't have blocked the nose. They die because they can't get the food to survive. And then okay, and then those people who are faster, it means the receptor is done properly. The affinity maturation, which I was talking, is happen very well. So these cells can recognize antigen faster than those who doesn't have affinity maturity. Comparing the key with the brush or without the brush. That's why those who had the brush, they will survive. And those who doesn't have it, they will die because they cannot get the antigen quickly and they will die. The positive signal is coming from the T cells, what happened here.